Welcome to Bewitching Brain. My name is Sophia Mazzacchio, and I am an MD candidate at the Medical College of Wisconsin. I'm super interested in the brain and nervous system as a whole. At the Medical College of Wisconsin, the Student Interest Group in Neurology provides educational outreach and is always looking for ways to engage community as we learn more about the bewitching brain. Please subscribe, share, and email me, or comment below with ideas or questions. Take good care, be safe, and help others be safe. The first part of this video will be focused on myelination, while the second part will focus on the different types of supporting cells of the nervous system. So let's get started with myelination. Myelination allows electrical signaling within neurons to be faster and more efficient. So, what actually is myelination? Myelination is essentially a process of insulating neurons, particularly axons, so that the action potential can spread more quickly toward the axon terminal. You can think of a myelinated axon as an insulated wire. The metal wire itself conducts electricity in one direction, while the coating speeds up the conduction by preventing leakage of the current into the air. Similarly, the axon conducts an action potential, while myelin prevents the current from leaking into the surrounding fluid. One important difference between electrical wire insulation and myelination is that myelin is composed of a fatty substance produced by other cells. We will discuss this further later in the video. Myelination is especially important for neurons that need to fire signals very rapidly. For instance, say that you are walking on your driveway with bare feet. You suddenly step on a nail, and your foot jerks upwards before you're even aware of what happened. This is a reflex, and it relies on myelinated sensory pain neurons to detect the nail and send a signal to your foot, and send a signal that your foot needs to move away quickly. Let's dive a little deeper into how myelination actually speeds up conduction of an action potential. The myelin does not cover the entire surface of the axon. Instead, it is divided into sections called myelin sheaths. In this illustration, you can see the axon with its myelin sheaths. The action potential will spread down the length of the axon all the way to the axon terminals. Here is a more zoomed-in depiction of a myelinated neuron. In between the myelin sheaths are small unmyelinated segments. These segments are called nodes of Ranvier. They have a high concentration of sodium channels. Recall that sodium channels allow for propagation of the action potential down the length of the axon. Sections that are myelinated allow for passive spread of the sodium ions to the next node of Ranvier. This type of conduction is called saltatory conduction because it seems as if the action potential is jumping from one node of Ranvier to the next. As a fun side note, the Spanish verb for to jump is saltar. Now that we understand how myelination functions to speed action potentials, and why that's important, let's talk about the supporting cells of the nervous system. Some supporting cells of the nervous system help neurons by myelinating them. Oligodendrocytes myelinate neurons in the central nervous system, while Schwann cells myelinate neurons in the peripheral nervous system. Recall that the central nervous system includes the brain and spinal cord, while the peripheral nervous system includes nerves outside of the central nervous system. Another important difference between these two types of myelinating cells is that one oligodendrocyte can myelinate multiple axons, while each Schwann cell only myelinates one axon. Here's a depiction of an oligodendrocyte and a Schwann cell. You can see that the oligodendrocyte has more than one process and is myelinating two different axons in the central nervous system. By contrast, the Schwann cells are both wrapped around only one axon in the peripheral nervous system. There are three other important types of supporting cells of the nervous system. Microglia have a few important jobs. They eliminate unneeded synapses, which is vital for learning. This is called synaptic pruning. They also clean up cellular debris in the nervous system and act as immune cells to get rid of bacteria or viruses that could harm the nervous system. Astrocytes have numerous functions that support the nervous system. One important function is to recycle neurotransmitters that would otherwise be toxic to neurons if they built up in large quantities. Another way in which astrocytes help the nervous system is by contributing to the blood-brain barrier. We will discuss the blood-brain barrier in a future video, but for now, just know that it prevents many substances in the blood from getting into the brain. This is necessary to protect the brain so that it is not affected or harmed by certain substances. The last type of supporting cell are ependymal cells. You can think of ependymal cells as a bathtub for the central nervous system. They create cerebrospinal fluid, which is analogous to water in a bathtub. They also contain the cerebrospinal fluid within the central nervous system. This is analogous to the walls of a bathtub. Here's a summary illustration of the supporting cells of the nervous system. You can see the microglial cell, which is cleaning cellular debris. You can also see the astrocyte, which is contributing to the blood-brain barrier and protecting the central nervous system neurons. It's amazing to think that all these cells are needed for proper functioning of the nervous system. I would like to extend a very special thanks to Dr. Teresa Patitucci, who graciously provided the images for this video. Dr. Patitucci is an incredible medical educator and artist. Thanks for listening, Brainiacs. Tune in next week for more.